Welcome back, everyone. This is the start of session 2, which is on contaminants, so plastics, environmental impacts and microbial interactions. I'm Martin Lilly. I work at DEFRA as a scientific advisor to the policy teams, so very much trying to bridge between the research that's going on here within Insight and then the policymakers and the things that they can do to intervene and potentially speak at OSPAR, as was mentioned by Colin earlier. Our first talk will be by Sarah Gow, looking at plastics and where they fit into pipelines and decommissioning. We'll then be followed by Natalie Hicks, looking at benthic impact, man-made structures, and Ben Gregson, thinking about the biodiversity around these decommissioned platforms. So if we can start with Sarah, please.
Hello. So yes, I'm going to be talking about legacy plastics from oil and gas installations, and that's specifically relating, in our case, to the pipelines and umbilicals. So this is, we're currently in our second Insight project. We were running on a slightly different model to the main projects. We had our first phase last year where we had a 3-month desk-based phase, which was kind of set in the background and trying to work out what the priorities were for understanding plastics and decommissioning. And we're about halfway through, or coming towards the end of our second phase, which will end in September. And we're trying to dig a bit more into the detail around plastics and think about how we need to consider them with decommissioning. So I wanted to just start by setting the scene a little bit in terms of where we're at with our thinking around plastics from a scientific point of view, and also what the policy landscape looks a little bit like, because I think that's important in understanding how we're going to consider this and decommissioning moving forward. So there's a lot of scientific consensus around the potential harm that plastics can cause. We know that they're found in all marine environments, right from the poles to the equators and the deep seas as well. There's records in Arctic sea ice and in deep sea trenches. And if we're thinking about how we classify plastics, we can think about the macroplastics, so that's the big stuff. Microplastics, which are the ones less than 5 millimeters in diameter. And we also think that there will be potentially nanoplastics. The scientific consensus is out on that at the moment because it's such a hard area to research. But that is something that's an emerging area, and there's research happening to try and identify if we can find nanoplastics in the environment. But it's reasonable to assume that they would be there. And if we think about this from a point of view of pipelines and umbilicals, then clearly they start as macro, but we would assume that over time, over the long time scales, they would be degrading down into micro and potentially nanoplastic particles as well. But we know, especially when we're thinking about oil and gas, plastics are really useful. They're essential in our operations. They're very versatile, durable and persistent, so they're great materials to use in subsea environments, but that's also what makes them really difficult to actually to kind of remove in the environment. So once they're in there, the environmental conditions subsea are such that it's very difficult for degradation to happen, and we don't yet understand whether they can be assimilated back into nature or mineralized. And by that we mean whether they would break down enough to return to their constituent elements and therefore not be able to cause harm anymore. So there's lots that we do know about the potential for harm now, especially from the larger plastics and microplastics. There's lots of evidence that it can cause ecological harm, whether that's through ingestion or entanglement, and potential bioaccumulation of plastics within tissues, and also toxicological effects, so the chemicals associated with plastics leaching from them. And we've now found evidence that that is not only potentially harmful in the marine environment to marine life, but also to human health as well. There's evidence of plastics being found in things like human blood and placentas. There's also the potential for lots of economic impacts as well, loss to tourism revenue and fisheries impacts. And if we look at the picture up of the beach there's a lot of research that's been done which shows the well-being benefits that come from us spending time in the marine environment. And if we've got beaches that are covered in debris, then clearly that has detrimental effects on those benefits as well. And if we put this into the context of where we are with policy, at the moment there is definitely scientific consensus that we've got, if emissions of plastic were to remain as they are at the moment under business as usual scenarios, we might be reaching the point where within the next century we get to ecological levels of things like microplastic concentrations in particular, where they're going to be tipping over into levels that we would know would cause biological harm. So at the moment, especially with microplastics, the studies that have been done are mainly lab-based and using elevated concentrations of plastic to look at the potential harm that they could cause. The ecological levels, what's in the environment at the moment, hasn't yet reached that level, but there is scope for it to do so relatively quickly if we do continue under business as usual scenarios. So it's clear, really, from a scientific perspective, that we should be taking action to reduce, prevent and mitigate plastic pollution in the marine environment. And this is a fundamental driver of the UN Plastics Treaty, which is under development at the moment, and it's going to be hopefully completed by the end of this year. We're going to reach some agreement on it by the end of this year, and it's likely to to change the way that we address plastics, right from the development and the design of plastics right through to their end of life as well. So it's something that we need to be thinking about when we're thinking about decommissioning decisions in terms of how that policy landscape is shifting. So how is this all relevant to oil and gas? So we know plastics are fundamental. They're essential components of pipelines and umbilicals. They're used for protection and for insulation. And as such, they're kind of essential to energy security. We need them. But we do have decisions that can be made around decommissioning. So if they are all left in the marine environment, if we decommission in situ, then what are the risks of doing that? And how should they be considered in decommissioning? How do we incorporate that, our understanding of their potential impacts, especially in light of changing political environment as well? So overall, our project aim is to determine how consideration of the risk from plastic associated with oil and gas pipelines and umbilicals should be incorporated into future policy and planning for decommissioning. So we've got some priority questions that we are addressing as part of our second phase. First of all, we're trying to work out how much plastic is there, how much plastic is contained within oil and gas pipelines. Because they were put into the environment such a long time ago, the ones that are now being decommissioned, we don't have easy access to information about what polymers they contain and how much quantity of plastic might be there. So we're trying to get some better understanding of that. We're also trying to improve our understanding of timescales for degradation and how long these things might take to break down into microplastics and whether they might actually mineralise it. What do we know about that and what might the timescales be there? We're also looking at what the environmental risks are from those plastics in terms of their potential to cause harm from an ecological and also socioeconomic perspective and the impact of any chemical leachates. And then trying to build all this into how we actually think about this in risk assessments and decision making and what are the criteria that we need to be aware of in order for industry and policymakers and regulators to take this and actually think about how plastics should be set against other criteria. Clearly, it's not an easy decision. There's a huge amount that needs to go into decommissioning decision-making, and plastic is only a small part of that. But how do we make it more visible, and how do we consider it? So in our first phase, we did some estimation of the amount of plastic that is contained within pipelines and umbilicals, and this work was done for us by John Gillies from ExxonMobil, and he did some estimations based on data from the North Sea Transition Authority and understanding of what pipelines look like and what plastics might be within them. So from that, we've estimated that there might be approximately 120,000 tonnes of plastics contained within pipelines. 80,000 tonnes of that may be decommissioned in situ, and that is because we know that the pipelines that it is contained within are the ones that are hardest to remove. They're rigid pipelines for the most part, or they're buried, and that makes the decommissioning harder. It's likely that we would be choosing to leave those in situ, and a further 40,000 tonnes in umbilicals, which we would assume would be removed from the environment because that's a bit easier. So at the moment, we're working with John to try and work out if we can be more accurate with our estimates. There's some work that's been done as part of the EcoStar project at St Andrews by Maria Martins, where they've done an excellent database of all the pipelines that are present in the North Sea. And we're using that to try and improve our estimates. And we've also been lucky enough to get some samples through Shell of pipelines from the Brent area of the North Sea, and you can see a picture of that on the slide there. That's a cross-section through the pipeline, so we can use the information in that about what plastic we can see in there to work out a little bit better to try and improve those estimates so we can try and improve those estimates. So in our first phase as well, we used some work that's been done in Australia. This is work produced by Curtin University, and they have identified in Australian context what polymers are contained within pipelines and looked at modelling of different environmental parameters to look at degradation timescales. And it's clear that degradation will happen on a very long timescale. We think it'll be hundreds of thousands of years before these plastics might be microplastics and might potentially, whether we'd get to that point of mineralisation. So we are going to be looking a bit more in depth at this data and using environmental data from the UK situation to try and see whether these are realistic estimates for the UK as well. And also with the samples that I mentioned that we've got from the North Sea, we're going to be doing some work about chemical weathering to see what we can identify. We've started this work, but it's quite early days with it, so I haven't got any results to present to you at the moment, but we've got samples that have come either from pipelines that are laying on the seabed, so exposed to the water column, partially covered with concrete mattresses or buried in the sediment, and we want to have a look and see if we can find evidence of chemical weathering that has occurred and how that might differ. And we can also use that then to critique those modelled timescales a little bit more. But when we're thinking about the environmental risks, I think we can all agree from the scientific consensus so far that there is potential for plastics associated with oil and gas, pipelines and umbilicals to cause harm, but it's how we actually frame that. So it's really difficult to take a fully risk-based approach to assessing risk in this way. If you take the example here, the plastic bottle, a whole plastic bottle might have harm in terms of economic and human well-being harm, but it's going to struggle to do harm to creatures unless they're really big, because they won't be able to ingest it. Once it's broken down into microplastics, then that's a different story. Then there's a much greater potential for harm to happen. And then once it's smaller and it can be assimilated by things like this scallop, then again, there's an even greater range of potential harm. And that includes economic harm through things like fisheries and also, which I think is quite important, public perception. If we start linking public perception and things like plastic being in fish and shellfish that we're eating, then that might have quite wide-ranging consequences. So we need to try and work out how plastics fit in and what it is, how much data do we need to get and how far do we need to go with our risk assessments before we can start making decisions. And at the moment, we don't have the evidence base that we probably need, but we need to try and work out what that should look like. There's also the concept of essentiality, and this is something that is likely to be incorporated in the Plastics Treaty. So this is when we can look at items that are made of plastic and work out if they're essential in terms of for health, safety, and safety of society. So if we take the example of a plastic drinking straw, that's clearly not essential, and we've already seen that they've been banned, and we've moved on to things like metal and cardboard instead. But it's a very different story when we're thinking about pipelines. Clearly they are essential, but the key thing here is that they're essential while they're in use. And once they're no longer in use, so if we decommission in situ, then they start to fall into the category of being abandoned, and that's in the definition of marine litter. And that's where we might start to have things happening around if they're classified as litter, should we be removing them from the environment? And also that's where the public perception comes in, I think, in terms of if the public were to think more broadly about this subject, then we might be in the situation where we see some public pressure in order to be making these, in order to be thinking a bit more about decommissioning and taking things out as well. So all of this is framing our thinking around how we're going to move forward. At the moment, we know that the processes of comparative analysis and environmental appraisal don't consider plastic that much. Clearly it would fit into quite a few of these principles, but in particular the environmental appraisal, it's hard at the moment because of the lack of robust evidence to make decisions on plastic. So hopefully the work that we're doing will be feeding into making that easier. But one of the key things that we're trying to do with this project is to actually formulate some decision-making support criteria. So using the research that we're doing, and then also with consultation with key stakeholders, so industry regulators and policymakers in particular, what do they need to know in order to consider plastics in decommissioning? What are the evidence gaps that they think are most important to fill in terms of shaping policy? And at the moment, we're starting that process. And if anyone here is interested in being involved, then we'd love to hear from you. So please reach out to one of us over the next couple of days, and we'd like to include you. We do have some future questions as well that we think are going to be key to addressing for this. So what can be done to minimise environmental accumulation of plastics? Questions around, can we design better? We know that we're going to be putting more pipelines and umbilicals in for oil and gas, but also more widely for other types of infrastructure. So for wind farms and everything like that, all of those cables and pipelines will be containing plastics. Can we do better in design in terms of being able to take them out again easily, or in terms of them not being potentially so damaging in terms of what plastic is made of? Are there options for compensation and mitigation in relation to materials that are having to be left in situ? And what's the scope for recycling? So we know that recycling does happen following decommissioning, but what does that really look like, and how easy is it, and how effective is it? What actually happens to this if it is removed and then taken for recycling? So hopefully this will form some of our thinking around the decision-making support criteria as well, and also hopefully into the future a bit more research. Okay, I forgot to say at the beginning that Richard, Thompson and Freya Mendrick are also part of the team, and they're here today as well. And we're also working with a chemical expert in plastic degradation from America, Anthony Andrade. So if you want to talk to one of us, then Richard and Freya and myself are here for the next 2 days. So thank you.
Yeah, thank you, Sarah. It's clear that the argument for leaving or removing is not clear, that there are multiple sort of nuances there, and it'll be interesting to see how your findings come out of that. Can we move on to Natalie next, please? So this will be about the benthic impacts of marine structures. Thank you.
So I'll be taking us a little bit away from what we talked about in the first session, where we looked at what was growing on the structures, and our project's really interested in understanding what the impacts are on the seabed itself. And so I'll be talking primarily about our NERC project FewComs, which is funded under Insight 2, and this is really focused on the oil and gas activity, but we're also interested in the renewables aspect, and we secured some additional funding under the same program, and that's the Orbit project, and so I'll talk a little bit about that too. So nevertheless, irrespective of which project we're talking about, the aim is pretty much the same. So we want to understand or determine the impact of these man-made structures, whether it's the presence, the placement, or the removal of them on the seabed, which is the large habitat around it. And we approach this in two different ways. So we've got a sort of large data analysis component within the FewComs project, and the view for that was to mine the existing data sets that have already been collected by industry over a number of decades, and also, of course, the collection of our own samples, so using a fieldwork and lab analysis campaign. And this is one of the challenges of working with industry data. Often it's being collected for survey purposes and not for scientific methodologies or for scientific questions in mind. And so we wanted to have some control over that by going out into the field and collecting our own data in a sort of scientific, robust way. Our funding actually arrived during the first lockdown, and as a very fieldwork-focused project, we had to really quickly think, and so we swapped our focus onto looking at the data initially. So I'll present some of that first. This is a paper that's just come out from one of our PhD students, Zelen Chen, and he was really interested in understanding the changes in species diversity around oil and gas platforms, and he wanted to see how that changed in terms of the food web and whether we can pick that up from existing industry data. So Zelen spent a significant amount of time looking at a number of different data sets, including UK benthos and some of the North Sea Transition Authority data sets, to get different species data. Each one of those species was assigned a trophic level, and then also looking at some of the environmental parameters that help us understand what some of those trends might mean. So he got 9 platforms going through this data set to find ones that had species data pre-construction and post-construction, but also the corresponding environmental parameters. And of course, since this data wasn't collected for scientific purposes, Zelen put his own experimental design on that data, which you can see here. So he used the hydrocarbon contaminations, or the concentration of hydrocarbons in the sediment, to impose his own regimes on them. So at the bottom here, we've got increasing distance away from these structures. And you can see the blue and the red line represent the concentrations of hydrocarbons in the sediment, and the blue is pre-construction, and the red is post-construction. So many of these platforms are still active at the time that the analyses were taken. And so he decided that between the 0 to 500 meter zone would be the impact zone, anything between 500 and 1500 is the buffer zone, and anything above 1500 is the sort of control zone, so what we're comparing things to. I'm just going to give a brief summary of what it is. There's a huge amount of species metrics and food web metrics within this, so if you are interested, please do go away and have a look at his paper in more detail. But in general, we're seeing within that impact zone, within the 500 meters of the platform, that we're seeing quite different food webs. We're seeing different species. There's a lot more opportunistic species. There's less diversity. And because there's less species and less trophic levels, we've got a more connected, simple food web. We also have an absence of many of the predator species. And as we move away into that buffer zone, we can see that start to change. We've got an increased diversity of the organisms within the sediment. We've got a more connected food web. Some of the higher trophic levels are coming back in. And then as we move into the control zone, it's pretty much what we'd expect for benthic food web. So that's some of the work Zelen's been doing, and he's now looking at that same data to see what the implications of decommissioning might have on the same metrics. And then we had fieldwork campaigns, which formed a core part of our project. And so I'll talk firstly about the work we've done at DEFUCOMS, which is looking at decommissioned oil and gas platforms. And then I'll go on to some of the renewables work. Those of you who are familiar with our project, and at the same time that Dan did his work earlier, and I know Fishpans as well use the same sites, we focused our sediment sampling around 2 decommissioned platforms in the northern North Sea. So Northwest Hutton, which is a bit further to the north, sort of halfway between Shetland and Norway, and Miller Platform, which is a little bit further south. They both decommissioned at slightly different times. So Northwest Hutton is the older one that's been decommissioned. We collected these sediment samples in 2021, so over a decade after decommissioning, and Miller Platform decommissioning for that platform finished in 2018. So it's a lot newer site. And we sampled the sediment at northerly and southerly directions for both of those, 2 gradients, starting at 50 meters close to the platform and doubling that distance as we moved away, so 100, 200, and so on, up to 3.2 kilometers away, which became our control site. And we're using these sediment data, we use them for analyzing pretty much everything we can think of. I think some collaborator called us milking everything we could out of the samples, and that's very much the approach we've taken. So biodiversity is one of the key things we wanted to really assess, and I'll present some of the macrofaunal data today. Ben will be talking after me, and he'll talk a bit more about some of the microbiology and the eDNA, but also some of the chemistry linked to that too. So there's a lot of talk for eDNA, but we believe as well we need to have those traditional approaches, so using the taxonomy to pick through the samples and see what's actually there before we can look at whether a genomics approach would align quite nicely with that. This is work that was led by Dave Patterson's group at St Andrews, and one of his members of the team has spent hours, days, weeks picking through the different samples, sieving through them to see what's there. And I'll start by showing you some of the changes of the relative abundance of the main genera, the macrofauna genera. This is platform Miller. So along the x-axis at the bottom, we've got increasing distance moving away from the platform, and the different colors represent different organisms, the different genus at this time, and the extent of that color shows the relative abundance. And we can see there's slightly different communities closer to the structure. It's a bit different to what we see as we move away, and as we move away from the structure, those communities become more similar to what we see at the control site. So for example, Echinocardium is in lower relative abundance quite close to the platform, and we know that's linked to its tolerance, tends to be relatively intolerant of pollution through metals or hydrocarbons. And the same we see with the Polychaete ceratulus. And in fact, the blue you can pick out really clearly is present at 50 meters, a little bit present still at 100 meters, and then disappears as we move away from the platform. And when we look at the same data for Northwest Hutton, we can see a similar trend. We've got slightly different colors representing different species, but closer to the platform, we've got a different community at 50 meters compared to as we move further away. And as we move further away, some of those colors shift. And as we get closer to that control site, we see the structure of the macrofaunal biodiversity starts to return to what we deem background levels. Of course, some of this is underpinned by what's happening in the geochemistry as well, and Ben will talk a bit about that, and it links to sediment types too. So these are just images taken from some of those samples that Mel was able to sieve through. But of course, we're really interested in understanding other things that the seabed provides, and we do a lot of work on blue carbon or carbon sequestration and storage, and one of our PhD students, Hugo, has been focusing his efforts on understanding how that might change around some of these man-made structures. When we wrote the project 5 years ago, we had the idealistic view of getting into active platforms when they still have that 500-meter exclusion zone. So we don't have those samples, but we do have some that have come out of after decommissioning. So we're kind of disentangling what's the result of decommissioning and what might be the actual background levels. So Hugo's paper is under review at the moment, and he quantified the carbon stock, so he looked at how much carbon there was. We used radiochemistry to estimate burial rates as well, but I won't be presenting that today. And so starting with Miller, again, we've got the increasing gradient moving away from the platform starting at 50 meters. The different colors represent the 2 transects, and you can see they're relatively similar. We've got an increase in the total carbon stock closer to the structure, and then that drops to relatively low levels, which is consistent across that gradient. And when we have a look at the same data for Northwest Hutton, we can see there's a lot more carbon within it. It appears to be a little bit more consistent. There's some variability at 200 and 400 meters, but nevertheless, we've got quite different carbon dynamics between the 2 sites. And some of that is driven by what's happening at the sites and what the type of sediment is. So Hugo had a look at what the sediment's made up of. So mud and sand are 2 terms that benthic biogeochemists love, and for us it's very important. I realize for most people that proportion of mud and sand is not quite so important. but it does have a correlation to what we'd expect to see in the carbon content. So this is Miller, and remember when I talked about the Miller carbon dynamics, we saw quite a lot of carbon stock quite close to the platform. That's partly driven by the finer particles, so we've got more mud content closer at 50 meters, and then it drops back to really small amount of mud, and it's basically a sandy sediment side. And so when you look at the sand content, which is in the lower plot, you can see it's pretty much the mirror image of what we see in the mud. So it's a sandier, more permeable sediment, and that pretty much represents what we have on most of our continental shelf sediments. Northwest Hutton was different. It was a nice muddier site. That's this perfect kind of mud that our group liked to work with. But nevertheless, we do see a slight trend, and closer to the platform, it's a little bit muddier than the background levels before returning to pretty much a flat line and what we'd expect. Okay, I'm not going to talk anymore about the decommissioned stuff, and Ben's going to pick this up in the next talk. But I'd like to chat a little bit about some of the work we've been doing in active wind farms too. So working closely with Ørsted, we swapped our focus to looking at renewables. Brilliant, thank you. And so we went out October last year after about a year or so of wrangling around different vessel options and how to get into the wind farm and what we're going to be using and deploying, etc. We went out in October last year. This time we were on board the vessel ourselves, collecting our own sediment samples. We were able to take a sediment profile imager with us as well, which is this nice bit of kit in the middle, belongs to CFAS. And that's a camera system that partially penetrates the seabed and takes a photo, so you get a nice image showing you what the sediment looks like, what animals might be living there. It tells you a little bit about the redox as well, and just some really nice visuals. Excitedly for us, we were given access to go within the farm, and so we were able to go inside the farm and sample there. So we have 3 sites within the farm, and then we sampled at transects away from the structure as well. We had an idealized 3 gradients, which we didn't quite get, but that's fine. So we've got inside 50 meters away from the footprint of the farm, 400 meters, and then a reference site again at 3.2 kilometers. And we thought this would give us a little bit of data to start comparing the difference between the impacts of oil and gas, where we can see hydrocarbons have quite a big effect on what we're seeing in the seabed, and also offshore wind, where we don't have quite that same hydrocarbon contaminants. Once again, we are still in the process of milking those samples for pretty much all the data we can. Biodiversity, again, is a big aspect, so I'll talk you through some of the initial macrofauna results that have come from Dave's group again. The microbial aspect will be covered a bit more detail this afternoon in Corinne's talk. And of course, the geochemistry, the metals are really important for understanding around wind farms too. We also were able to bring some sediment back in the lab, and we set up a small mesocosm experiment to control the conditions and the types of metals added to sediments to see how that influences some of the services, such as the remediation driven by the microbial communities. So in terms of what macrofauna we're finding at the site, so Barrow is one of the oldest wind farms in the UK. It's about 20 years now since it's been commissioned, so we'd expect to see some changes or perhaps a return to what it was pre-construction. These are some of the images that Mel's produced from her sample. She's probably about halfway through. She's got another half to sort through. Days at the microscope. She's got a lot more patience than I do, but also a skill for doing it. And we can see there's quite a wide diversity in the different organisms that we're seeing. These are also taken from Barrow samples, and these are images from our bioimaging facility at Essex. I think they're quite beautiful for seeing some of the detail we don't often see on the species. I particularly like the tube worm on the far left, where you can see the individual sediment particles in the tube. I think that's pretty cool. Anyway, irrespective of that, we are starting to be able to pick out some trends. So we can see that Amphiorofiliformis is really dominant species here. When we look back at the pre-construction and post-construction surveys, we can see that that was present pre-construction and not in the post-construction survey. So whether that indicates perhaps they're returning to the area, the environment's maybe gone back the same, maybe the sediments changed a little bit. We're seeing some dominant other species as well. What we are clearly seeing are differences in the sediment type too, and so we're just unpicking at the moment whether that's what it always has been or if that's a result of having the structures there as well. So there's a lot more work to go through on this, and if you are interested, please do come and see Corinne's talk. She'll be presenting some of the work Hayley, our PhD student, has been doing on the microbial aspect too. And of course, we've got all this data, and it's not just an academic output. We're looking at using that, using different models or tools to sort of expand that. Chris will talk a bit later about some of the food web work he's doing, but we're also looking at plugging some of this data into models that already exist. So the spatially managed marine area toolkit, which is the smart spatial tool that CFAs have. And that allows you to sort of predict what impacts might have on certain aspects of the seabed. So this is a recent DEFRA report that came out, and what you can see here, the different colors on the seabed itself are the carbon vulnerability, and you can use it for things like carbon sequestration, biodiversity. And then the little cutout sort of diagonal things are red for MPAs, marine protected areas, and the pinks are the offshore wind. And so effectively, if you know how the seabed is changing in response to something such as the presence of an offshore wind farm, what that means for the biodiversity, you can then start to layer that into the model and say, okay, if we put a wind farm here, how does that affect biodiversity? If we move over here, do we see changes in carbon sequestration? And that has been used recently for looking at where we put offshore wind farms, how that impacts catch as well. So there's an economic linkers there. So we've done quite a lot of work so far. We've still got quite a bit to come, so please watch this space. Happy to take any questions on email if anyone doesn't want to ask them today. These are the names of all the academic collaborators that have been involved, but we've been hugely supported by a number of industry partners too. So thank you very much.
Thank you, Natalie. Really interesting to see the opportunities you can get from the offshore wind and then from the oil and gas, and whether the results are the same. If I can invite Ben to come up, please.
So, hello everyone. Oh, can you hear me? Yeah, cool. Hello everyone, I'm Ben Gregson, a molecular ecologist, currently based at Anglia Ruskin University, but I did work on the FewComs project for just under a year. As a molecular ecologist, I use a combination of environmental DNA and DNA sequencing approaches to determine the biodiversity and function within environmental samples. So as part of the FewComs project, we've used those approaches to see what the impact of decommissioned oil and gas platforms have on the biodiversity on the seabed surrounding these decommissioned structures. Just so we're all kind of on the same page, as I see the term used a lot incorrectly, eDNA is defined as DNA that we obtain from environmental samples. That can be water, air, sediment, and it's from the sample rather than sample directly from an individual. So as an example, some of the sources can include feces, shed skin cells, microbial cells present in the environmental sample, basically anything that's getting released from an organism into that environment. So that it's essentially acting as a genetic imprint that that organism has been present or is present within that environmental sample. So as an example, I'd get eDNA from the water that a fish has swum in, not directly from the fish itself. And once you've extracted eDNA from environmental samples, there's two different ways you can kind of use it. You can use it to target individual species, so in freshwater ecosystems, if they want to monitor invasive species like crayfish or detect rare species like the great crested newt, they'll specifically target the eDNA released by those species. eDNA is also very useful to distinguish between very cryptic species. You can have two seahorses by each other that look like mirror images of each other, but they're distinct species, so eDNA provides a tool where you can actually distinguish between those. But where the real power of eDNA comes in is where you're able to characterize multiple organisms at the same time. So that's everything from the microbes across all domains of life, microbes, archaea, fungi, plants. So you've got the opportunity to survey an entire community to properly capture the biodiversity present within an environment. And eDNA has a lot of advantages over the kind of traditional visual surveys. So for example, you don't need a fish taxonomist to come, you don't need a algal taxonomist, you don't need lots of experts with taxonomic expertise. All you need to do is collect the environmental sample, extract it in the lab, and then a computer is going to be doing most of the heavy lifting in terms of the taxonomic identification. eDNA is a lot more sensitive, so you can detect species that are either very rare or that can't be seen. So for example, microbes. It's a lot more accurate because you're able to characterize that entire community, so you get a much more accurate overview of the biodiversity present. And also most sampling techniques are non-invasive, so you're either not disrupting the environment or having direct contact with the organism of interest. So as Nat kind of described, we have two decommissioned oil and gas platforms, Miller and Northwest Hutton. We've collected sediment samples at increasing sampling distances, 50, 100, doubling sampling distances up to 3,200 meters. We've extracted eDNA from those sediments, and then we've used a variety of different taxonomic marker genes to characterize the communities that are there. You'll use different taxonomic marker genes to target different taxonomic groups. So if I'm interested in microbes, I target the 16S gene. If I'm interested in eukaryotes, I target the 18S gene. So specifically for this project, we've targeted the 16S gene to characterize the archaeal and bacterial communities. And we've done the 18S to characterize the eukaryotic communities, and the ITS gene to characterize the fungal communities. For the sake of time today, and some of the data hasn't been fully analyzed, I'm only going to be focusing on the microbial component, so just the 16S data. So after we've amplified up that taxonomic marker gene of interest, we then use a DNA sequencer, so we can determine the order of the nucleotide bases, the order of the G, Ts, As, and Cs. Once we've got that, we can then assign taxonomy to those sequences. So at the end of it, we have a list of species that are present in our environmental sample, and we have an idea of the relative abundances of those organisms. We also chemically characterize the sediments, so we're interested in kind of if we see any differences in the biodiversity, what are some of the environmental variables that are driving those distances? So we characterize some pollutants, some heavy metals through ICP-MS, different hydrocarbons associated with the oil through GCMS, so a range of different aliphatic and aromatic hydrocarbons. We're interested in the nutrient cycling being conducted by the microbes, so we did a bit of ion chromatography to determine the concentration of different anions and cations, and also some more general physicochemical characteristics of the sediment. Nat mentioned the carbon content, but we also measured the pH of the sediments. So first, here you can see the top, you have the total alkane concentration. On the x-axis, you have the sampling distances. On the left, you have the results from the Miller platform. On the right, you have the results of the Northwest Hutton platform. And you can see, unsurprisingly, as you get closer to these decommissioned oil and gas platforms, you have very high concentrations of both alkanes and aromatic, polycyclic aromatic hydrocarbons. And Northwest Hutton was obviously decommissioned about 15 years ago, but you're still seeing very high levels of the aromatic hydrocarbons. And when you actually so these are total values, so if you actually break it into the individual compounds, you'll see for the alkanes, this is mainly driven by hydrocarbons that are 20 carbon chains in length and longer, so kind of the longer, more recalcitrant alkanes. And then for the polycyclic aromatic hydrocarbons, it's more 3 and 4 ringed structures, so really difficult to break down, so the compounds that are very difficult for the microbes to break down. Moving on to the heavy metal data. So again, similar structure, you've got sampling distance on the x-axis, heavy metal concentration on the y-axis, and the dashed line represent different concentration thresholds from the Environmental Protection Agency. So MP stands for moderately polluted, HP stands for heavily polluted, and you can see again, the sediments collected closest to the decommissioned platforms have very high levels of heavy metals. So for example, it's moderately polluted with chromium, iron, and nickel, and it's also heavily polluted with copper and zinc. Yeah, and also at Northwest Hutton, heavily polluted with lead. We couldn't detect any lead at Miller, but I think that's probably a mistake in the methodology. Main result from the ion data is we saw very high levels of sulfate at the Northwest Hutton platform. So As you saw actual gradients, as you moved further away from the decommissioned platform, you saw the levels of sulfate significantly reduce, with the highest levels being at those 50-metre sediments. When we look at the Miller platform, there actually was no significant difference in sulfate concentration, and they're all slightly lower, but no differences between the sampling distances. So for the pH, again specifically at Northwest Hutton, we saw that the sediments closest to the platform were more neutral, and as you moved further away, they became slightly more acidic, so decreasing from a pH of around 6.25 to 5.8. And similar story to the sulfate data, there wasn't actually any significant difference in the pH at the Miller platform. So now moving on to the DNA sequencing data. So this is the bacterial results. Image on the left just shows different alpha diversity metrics. They take into account things such as the number of species detected and the relative abundance of different species, but regardless of which alpha diversity index was used, we could see that the bacterial diversity is always lowest at the 50-metre sediments compared to the greater sampling distances, and that was consistent for both the Miller and the Northwest Hutton platform. And we think this is likely due to selection by the high level of pollutants that we've seen, the hydrocarbons and the heavy metals. This shows the difference in the composition of the bacterial community. So each dot represents an entire bacterial community for a sample. Closer they are together, the more similar they are. The further apart they are, the more distinct they are. And we can see that there's a significant difference in the bacterial community composition based on sampling distances. So you can see the ones in blue, that shows that the bacterial communities in the 50-metre sediments are distinct compared to those later sampling distances. And as sampling distance increases, the communities become more and more similar, and you see the dots start to converge. So now let's actually look who is there, what's the actual composition look like. So you have on the y-axis the relative abundance of, so here we're representing the most abundant bacterial genera detected in our samples, and we can see that the microbial communities are dominated by sulfur cycling microbes. So for example, the microbe in yellow, sulforovum, that's a sulfur oxidizing bacteria, so converting sulfide to sulfate. And desulflatigans in the dark blue, that's a sulfur reducing bacteria doing the reverse reaction, converting sulfate to sulfide. So they're dominating at the 50-metre, in the 50-metre samples, and then as sampling distance increases, their relative abundance decrease. So now we know there are distinct differences in the communities. We're also interested what variables are driving those distances. At Miller, it seemed to be that most of the communities were driven by ammonium concentration, but as you can see, the dots aren't really separating very well, so that's not a very strong effect. Whereas at Northwest Hutton, the bacterial communities the differences in the bacterial communities has been strongly driven by those alkane concentrations and heavy metal concentrations, and also specifically the pH. So why are we seeing these sulfur cycling microbes at increased relative abundance? So first we have enhanced sulfur oxidation by that sulforovum genera, and from the literature we know that they're resistant to high levels of heavy metals and hydrocarbons, so they're dominating the microbial communities, and they're catalyzing the reaction of sulfide to sulfate. An increase in sulfate, which we see from the chemistry data, is stimulating the growth of sulfate-reducing bacteria, so that's leading to more sulfate reduction. That reaction reduces the amount of protons present, so that's why we're seeing an increase in the pH in those sediments, and then that's also going to have a knock-on effect to the retention of heavy metals in the sediment. Kind of similar story for the archaea. There wasn't really a significant difference at Miller, but there was at Northwest Hutton. And specifically, the only significant differences was between the 50-metre samples compared to ones greater than 200 metres. Again, looking at the archaeal composition, at Northwest Hutton, we see methanogens, so methane-producing archaea, which are making up over half of the community present in the 50-metre sediments. And it's specifically a hydrogenotrophic methanogen, so it uses hydrogen as an electron donor, and you can see it massively dominating the community. It's the one in light green. Similar story to the bacteria, not a strong effect at Miller, and also there's not really much of a difference in the communities, but again, hugely driven by the alkane and heavy metal concentrations. Why are the methanogens doing so well? So hydrogenotrophic methanogens do better at more neutral pHs. We know that the sediments are more neutral, so it gives them a chance to outcompete other microbes. Sulfides produced from the sulfate-reducing bacteria could precipitate with the metals forming with the metal sulfides, and then that reduces the negative effects that the sulfides and the heavy metals have on methanogen growth. And it's also possible that methanogenium is forming a syntrophic relationship with some anaerobic hydrocarbon-degrading bacteria. So this shows that we've done the 16S data, but we have plans to also characterize the eukaryotic and fungal communities. We're also kind of inferring function with this amplicon sequencing. We plan to do some shotgun metagenomic sequencing so we can characterize both the taxonomic and functional genes present within the environmental sample. And as you can see from Corin's talk, we're going to apply this approach to other man-made structures, such as wind turbines. So in summary, I hope this talk showed you kind of the power that eDNA has, and it can be a really useful tool for biomonitoring around decommissioned man-made structures. We're seeing some differences within the sulfur cycle and the carbon cycle in relation to decommissioning age. So it'd be interesting if we collected the samples from Miller at a later date. Would that then look the same as at Northwest Hutton? And I've also hopefully highlighted that microbial biodiversity is also important. Of course, macrofauna are also important, but I think we need to take a multi-domain approach where we're characterizing every domain of life, microbes, macrofauna, to fully assess the environmental impact of these structures. Thank you all for listening.
Thank you. Could I ask the speakers to take their places for the question and answer? We've got 10 or 12 minutes for that, so if there are any questions in the room, and will there be questions online, potentially? Yeah. So, can we have in the middle first and then at the back?
Hi, thanks. I'm Corinne Whitby from the University of Essex. This is a question for Sarah. So really good talk, Sarah. I was interested in, you mentioned doing some chemical weathering experiments, and I wondered if you could maybe elaborate a little bit more on that, and maybe ask you, are you considering doing some biological degradation experiments to maybe speed that process up?
Freya, who's in the audience, who just passed you the microphone actually, she is doing that work for us, so it might be good to pick her brains a little bit more about the chemical weathering element, but I can tell you that we are doing FTIR analysis to look at the polymer and how we've got the samples underneath, so the protected stuff that's been within the pipeline versus what's been exposed to the environment. We're doing comparisons between the two to see if there's differences in the polymer, and we can look at chemical weathering. We're also going to be doing some hardness testing and some hopefully some pyrolysis as well to look at leachates and things like that as well. In terms of the biology, the samples were pressure washed as they were removed from the environment, so there's not much that we could do there, but it's something that we'd be very interested in. And actually the microbial things as well, that's really interesting to us in terms of how the plastic might be being weathered by the biological community. So not at the moment, but it's something that would be good to investigate.
Hello, Anthony Knight, University College Cork. Question for Ben, actually. I'm not a molecular ecologist, so apologies for my naivety if this is not the case. I didn't think eDNA could measure abundance. I thought it was purely presence-absence. So could you clarify that for me? That's really...
Yeah, so kind of the output of the DNA sequencing data is you have counts for how often that particular DNA sequence occurs within your sample. And then from that, you can kind of calculate changes in the relative abundance, not absolute abundance. So you could alternatively use a different approach, qPCR, to get a rough idea of the total number of that gene, and then you could kind of apply the relative abundance number to that. So if I calculate 100 million 16S genes, and then I see the relative abundance is 40%, that could give me a rough estimation.
So how do you, have you validated that then? I mean, I've seen in other cases where they've done it for fish, for example, where they've actually gone out and sampled true numbers and correlated that to...
So as Nat showed, we've got the macrofauna data, and we're going to analyze the 18S data and then see if there's any parity or disparity between the two measures, basically.
Thanks.
Okay, if we can take the next question online, please.
Yes, great, thank you. We've got a question from David Santello. For Natalie, interesting approach to analysis of existing data. Have these analyses also been able to include meiofauna, and are meiofauna communities included alongside macrofauna and microbiota in the more recent studies?
For our project, no, we haven't looked at meiofauna. We could probably pick that up with the genetic approaches. But for Zeland's work, which I think is what he's maybe interested in, Zeland went through everything that was in the database, and there are some meiofauna there too. So if you have a look at his paper, for every species, he's assigned at different trophic level, and it's things from detritivores to filter feeders to predators. So yeah, some of that is in there, but meiofauna is a big gap, and I know within the UK we don't have a huge number of experts for identifying that, so it's definitely something that's missing.
Are there other? Right.
Yes, there's one here for Sarah. So what's the sort of relative contribution you think of, David Patterson, University of St Andrews, what do you think the relative contribution or likely relative contribution of plastics from degradation from oil and gas is compared to the amount that we're putting in from land and from other industrial processes? Thanks.
Yeah, it's a really good question. It's something we've been trying to consider, because obviously that is very relevant. It's not something we've managed to get actual numbers on yet, so we don't know what proportions we're talking about. But I think the overall point is that regardless of what the contribution is, it's still going to be plastic that is persisting in the environment for hundreds of thousands of years. So it's adding to that standing stock, and we know that the standing stock of plastics in the environment is just increasing. So I guess the focus is on trying to think about how we can reduce it on a global scale from all the sources. So, but yeah, we will be trying to put that into context with our report. We touched on it a little bit in the first phase, but it's quite difficult to quantify it. But yeah.
Thank you. Questions in the middle here, please.
Hi, I'm Jordan from the University of Hull. I have a question for Natalie. So obviously you said that the data that is often collected by industry doesn't benefit a lot of what we're trying to get out of it, but what do you think of seasonality in terms of the data that's collected by industry compared to what you're then getting? Is it matching or...?
Yeah, seasonality is a good point, and it's something we think about, and I think it depends what metrics you're measuring as well. So, for example, we measure carbon quite a lot, and while at depth that carbon might not change on a seasonal aspect, we know it does on the surface with blooms as well. That's not really something that has been captured. We know from Zeland's work that sometimes the time that data was collected has been included, other times it's just a year, and so yeah, we don't have a good handle on that. But you're right, for some things it'll be really important.
Thank you.
If we can take the next one online, and then in the front there, please.
Okay, thank you. Natalie, it's another one for you, I'm afraid. From Amit Jorgensen online. She's wondering if you were also able to compare the change in biodiversity and blue carbon pre and post construction with the situation after decommissioning. And it would be interesting to know what extent biodiversity recovers after decommissioning.
Yeah, I'm all on board for any more blue carbon work for sure. The difficulty with carbon measurements is it needs to be collected for carbon. So for things like the macrofaunal, you can grab and get a chunk of sediment. For carbon, you need that sediment profile, so you can look at the amount of carbon in those layers, but you also need that sedimentation, and we don't tend to have that with industry data too. So it is a bit of a gap, and for us personally, we'd love to see that change because the seabed's being changed into how it's used around these oil and gas platforms. You don't have things like trawling going in, so we think that's affecting some of the baseline dynamics, but that's a time snapshot we don't yet have, and we'd be really keen to get.
Hi, Josh Lawrence again from Heriot Watt. A question for Natalie and a bit for Ben. Something we've been struggling with a little bit in fish spams is the concept of a baseline or background levels, and sort of we're thinking about fish densities. But I was wondering how happy you are with 3.2 kilometers being far enough away to be completely outside the impact. Like there was on some of the plots you showed, like the echinoderms, it was increasing as you moved away and then maybe dropped off. And I think the same with the pH, it was dropping and then increased to the furthest distance. How can you sort of attribute that to the platform versus patchiness globally across the North Sea? And are there other data sources that you can use to try and establish a robust baseline?
Okay, I'll start and then feel free to jump in. Yeah, no, absolutely, you're right. And when we did this, we were trying to make sure we had a control site that represented the same hydrodynamic sediment type as well and would be far enough away from the platform to be outside of that footprint. One of the things we have done is we've used radiochemistry in the sediment samples, looking at different radionuclides. So we're looking at lead-210, cesium, and radium, and those are elevated and produce or discharge water from the platform. And we can actually measure how far that impact has gone around the platform. So we know for Northwest Hutton that's extended to about 200 meters. And then beyond that, we don't have the impact of the platform. So in reality, anything perhaps outside of that exclusion zone would be a good control. So that's been quite a useful and unintended aspect that we found during the project. Do you want to add anything? Yeah. Does that answer your question? I think that's the baseline thing is really difficult for sure. And of course, there's other things going on. As soon as you move out of that exclusion zone for an active site, you've got other activities happening on the seabed which are having that impact. And we're seeing some of that with the changes in biodiversity. So you're now into areas that are trawls, so the biodiversity is going to be monitored anyway. So it's almost like when Hugo talks about his work, it's almost like a halo effect. So beyond that 200 meters and before you get outside that 500 meters, you've almost got what we'd expect as baseline sea dynamics before that exclusion zone goes. So we're not having trawling, and we know the impact from the platform is not extending beyond that. Of course, that will be site-specific and depends on the hydrodynamics of the movement of water and how that's being moved as well. But yeah, it's really interesting, complex.
Right, we've got time for one more question before lunch, and David at the back.
It's Richard, actually. Richard Thompson,
Richard.
University of Plymouth. I wanted to partly chip in to Dave's question, which is a really good one about the quantities of plastic in oil and gas related to other sources, because it is, as Sarah said, something we've been looking at. And I think, so you could compare it, let's say, to the legislation that the UK introduced to prevent microbeads in rinse-off cosmetics, and the quantities of plastic associated with oil and gas way exceeds that as an area where we've seen legislation. It also far exceeds the quantities that have been picked up by a decade of beach cleans organised by Marine Conservation Society. So it's not an inconsiderable amount. I think it's important to also recognise that we're in an era when those bad practices in other industries are hopefully going to diminish. So the relative contribution of that from oil and gas is likely to potentially become more important. Now, those other forms, the microbeads in cosmetics and the litter, aren't essential things in the marine environment. And it's those things that we need to balance. But potentially part of the issue here is the optics of what is done. That if we're agreeing to leave in the ocean a quantity of plastic that far exceeds something that we've seen DEFRA legislate against, that might not make sense to everybody. So it's about trying to weigh one thing against the other and provide some of the comparators exactly along the lines you've been saying, to try and set it into context. So it's a really good question, and we'd welcome further thoughts on that, because it then boils down to this essentiality criteria, because clearly the two examples of bad practice I've given you are about non-essential things. Oil and gas pipelines are essential, so it's how do you weigh, until they're decommissioned and they're not used. So how do you weigh those things against each other? It's really good point. Thank you.
Well, it'll be good to see discussion on this continue, and please do grab the speakers at lunch. We'll be coming back at 1:30 for the session 3, which is biodiversity effects, food webs and species interactions. So, yes, lunch will be in the main area. Thank you.